Okay, it is my pleasure to introduce today's joint NIMH Center Series seminar speaker, Dr. Reina Bendayan, professor in the Department of Pharmaceutical Sciences at the Leslie Down Faculty of Pharmacy and Institute of Medical Sciences, Timothy Faculty of Medicine at the University of Toronto. After obtaining a Bachelor's of Sciences in Pharmacy and a Hospital Pharmacy Residency program at the University of Montreal, Dr. Bendayan completed a Doctor of Pharmacy at the University of Florida and a 3-year Medical Research Council postdoctoral fellow program in clinical pharmacology and membrane cell biology at the University of Toronto. And I guess she decided to stay in Toronto. So her current research program is primarily focused on membrane transport and therapeutics with the emphasis in the field of HIV/AIDS, antiviral drug transport, and regulation at blood tissue barriers, which are considered as the sanctuary sites and cellular reservoirs of HIV virus. She's also over more than 120 peer-reviewed manuscripts and has supervised many graduate students and postdoctoral research fellows. She was an elected fellow of several scientific societies and the recipient of the Association of Faculties of Pharmacy of Canada Research Career Award, the AAPS Award, and the CSPS Leadership Award. The title of her talk today is Uncovering a Novel Molecular Therapeutic Target for HIV-Associated Neurocognitive Impairment. So I'd like to turn this over to Dr. Bendayan.
Thank you very much for the kind introduction. Hello, everyone. It's a pleasure being here. It's actually, I'm coming back. I was mentioning that I presented on similar topic pre-COVID in 2019. And then everything got interrupted in March 2020 with the closure of the laboratories. And as many, we lost a number of our HIV, EcoHIV mouse model, and other transgenic models for a number of our studies. So that kind of put us behind, but since a few years, we came back and moved on with a similar topic that I discussed a few years, as I said, in 2019, addressing novel molecular therapeutic targets for HIV-associated neurocognitive impairment. So the work is done at the University of Toronto at the Leslie Down Faculty of Pharmacy. We are located in this very nice, relatively new facility, which is really at the center of our downtown St. George campus. I want to acknowledge a number of members, contributing members of my laboratory that have participated to this work, in particular, doctoral students, Tamima Ashraf, Amila Omerajik, and our senior research associate, Toza Melhok, and our present doctoral candidate, Celine Titus, that is pursuing this work. So as you know, the HIV epidemics is continuing to rise, with close to 40 million people worldwide living with HIV. And despite the discovery of very effective pharmacotherapies when combined that maintain very low viral loads or eradicate the viral loads in the periphery, there is no cure at present for HIV, as you know. Some of the reasons for that, obviously, are the fact that the virus mutates constantly, but also in part also due to the fact that the virus can persist in a number of cellular reservoirs, primarily the resting T cells, and also continue to replicate in a number of viral sanctuaries, tissue sanctuaries. And these tissues are highly protected by blood tissues barriers, particularly the brain, the male genital tract, particularly the testes, that prevent effective permeability of a number of anti-HIV drugs to reach these tissues. And our laboratory primarily has had a lot of interest in addressing these questions over the past many years, in particular in the brain, although we did recently a number of studies in the testes as well. And the reason for that, in part, is because a number of efflux pumps belonging to the ABC membrane transporters, ATP binding cassette membrane transporters, such as P-glycoprotein and breast cancer resistant proteins, are very effective in effluxing actively a number of these anti-HIV drugs, along with a number of many other substrates. Obviously, the characteristics of these barriers, in addition to the biochemical function, the physical characteristics in the brain of the microvessel endothelial cells being adjoined by very tight junctions, make it a very effective physical barrier with high electrical resistance also that contributes in preventing permeability and delivery of a number of therapeutics. And this is an overall challenge in the pharmaceutical sciences. HIV-associated neurocognitive impairment, along with HIV-associated inflammation, is a major problem in the HIV population. And as the population is aging and increasing, this complication is really prevailing and becoming a clinical problem in patients. So up to 50% of HIV-infected individuals can experience cognitive deficits, behavioral disorders such as anxiety, depression, as well as motor deficits. And at present, there is no treatment for this particular condition. So in addition of our interest in biodistribution and delivery of anti-HIV drugs to target sites, this has been an interest over the past several years. So although this group is very familiar with HIV neuropathogenesis, I thought that I will go over this aspect briefly, just to put us in context of the work that we have been doing. So it's important to indicate that very early on in the onset of HIV infection, the virus is able to penetrate into brain parenchyma and cause infection in the brain. So how this happens is how the virus can cross the blood-brain barrier, this very tight barrier, is through various pathways, ones being through infected monocytes from the periphery that can cross through paracellularly in between the cells, because here in the context of HIV, we are going to see a much leakier barrier due to the inflammation that is associated with the infection in the periphery. So therefore, those paracellular spaces are then available for delivery, for transport. So that is one way. In addition, the virus, the virion alone by themselves can also cross at the level of the paracellular spaces. And finally, transcellularly through transcytosis processes via potentially the mannose-6-phosphate receptor, the virus can gain access to brain parenchyma. Once in the brain, the major target are the microglia cells, which are the primary immune cells in the brain, as well as to a less extent the astrocytes. And at this level, it is now believed that there is non-productive infection. However, with the infection of both microglia and astrocytes, there is activation, and this allows for the release of a number of pro-inflammatory cytokines, chemokines, as well as reactive oxygen species that will form inflammation, a condition of inflammation in brain parenchyma. In addition, it's important to indicate that at the level of the astrocytes and also at the level of the neurons, but especially in astrocytes, there is high expression of glutamate transporters in the murine, the glutamate transporters, the GLUT1. In humans, the AT1 and 2 transporters play a very important role in this active reabsorption of glutamate. And these are dysregulated during the infection, and therefore we are having accumulation of the excitotoxin of glutamate in brain parenchyma. So together, this will lead to neuronal injury, which in the clinic will translate, is believed, to these cognitive, neurocognitive impairments that are seen. So I want to come back to the limitations of antiretroviral therapy. I mentioned the fact that many of the antiretroviral drugs don't permeate across the blood-brain barrier and reach effective concentrations in the brain due to not only the physical barrier, but primarily to the effect of the ATP binding cassette membrane transporters that actively efflux a number of these agents. And over the past many years, we've done a lot of this work demonstrating the subtherapeutic concentrations at the level of the brain, but also in testicular tissue. In addition to the poor permeability, most antiretroviral drugs don't have direct anti-inflammatory effects. Aside from Maraviroc, the CCR5 inhibitor, the others primarily don't exert anti-inflammatory effects, which are quite important in this context. Paradoxically, when some of the anti-HIV drugs are able to reach the brain, a number of them exert neurotoxicity. For example, efavirenz. We are also interested in this aspect and undertaking some research project, primarily looking at neurotoxicity of integrase inhibitors such as dolutegravir. And we've demonstrated that exposure of dolutegravir in vitro and in vivo causes brain inflammation and blood-brain barrier disruption, tight junction protein downregulation with increased permeability. So this is paradoxical, something that we need to keep in mind. So basically putting all of this together, then thinking about HIV-associated cognitive impairments, that there is a need for novel therapeutic approaches as adjuvant therapies to antiretroviral drugs. This is where we came across and became interested in the peroxisome proliferator activated receptor, in particular the PPAR gamma isoform. So this is a ligand activated transcription factor that plays a very important role in lipid and glucose metabolism, and where the specific agonists, the members of the thioglitazones, such as rosiglitazone and pioglitazones, are common drugs available in the clinic for the treatment of diabetes type 2. However, more recently, and this is quite interesting, treatment with PPAR gamma agonists, such as the thioglitazones, have demonstrated neuroprotective effects. And this is what has caught our attention. And what has been demonstrated is that the PPARs, in the context of neurological disorders, are able to mitigate and decrease microglia and astrocyte activation, resulting in mitochondrial bioenergetics, promoting mitochondrial bioenergetics, neuronal survival, as well as oligodendrocyte maturation, and decreasing T cell activation. At present, there is compelling evidence suggesting that the activation of these receptors, particularly the PPAR gamma, play a role in modulating or regulating inflammation in models of neurological disorders such as multiple sclerosis, Alzheimer's disease, traumatic brain injury. We could add Parkinson's disease as well. So how does the PPAR act in terms of the proposed mechanism? It is not very clear, but it has been proposed that it is through a trans repression of the NF-kB signaling pathway. So NF-kB is highly activated in the context of triggers such as infection and inflammation, and turn on the transcription of a number of pro-inflammatory cytokines, chemokines, reactive oxygen species, etc. So it has been proposed that the activation of the PPAR, let's say PPAR gamma, will inhibit the P65 subunit of the NF-kB that reaches the nucleus when the NF-kB pathway is activated and there is translocation into the nucleus. And it is at this level that it has been proposed to act as one of the pathways. And this is a pathway that we have examined, as I will describe in a moment. So now PPARs in the context of HIV, there has been some data and some evidence that they could play a role in the context of HIV infection of the brain. So some interesting, very early studies by the group of Yuri Perstisky at Temple University demonstrated that in the SCID mouse model of HIV encephalitis administer monocyte-derived macrophages derived from associated with HIV-ADA could lead to HIV replication in macrophages, and the administration of a PPAR gamma specific agonist such as rosiglitazone could mitigate these effects. So this was an earlier, very interesting initial study. Another study by the group of Michael Toporek, this was done when he was based at the University of Kentucky. He more recently, pre-COVID, moved to Miami. But this study was done in wild-type mouse, administered TAT intracerebrally, demonstrating that the viral protein could lead to astrogliosis in the brain, neuronal loss, disruption of tight junction proteins with downregulation of those, with disruption of blood-brain barrier integrity. In the group of mice receiving either a PPAR alpha or a PPAR gamma agonist, there could be also mitigation of these effects. So these are interesting initial studies alluding to the fact that the PPARs could have a role to play in the context of HIV infection of the brain and associated inflammation. So this, in terms, goes to the rationale of our work, ties in with it, the fact that HAND, or as it's called now, NCI, is a complication that is on the rise, especially as we think about HIV population living much longer and aging. This becomes a problem predominant in the clinic that affects the quality of life of individuals living with HIV. The fact that ARV therapy is not that effective, not only for having low brain permeability, but also potentially for some of the antiretroviral drugs of being neurotoxic. So therefore, our rational and related question has been, can PPAR be a molecular target for HIV-associated inflammation and infection in the brain, and could some of the agonists be of interest? So our studies started with a very simple model, and I'm only showing in the interest of time, some data in vivo, where we did data in vitro in primary cultures of astrocytes. We also work with primary cultures of mixed microglia and astrocytes. In this model, we only injected the envelope glycoprotein HIV-120 intracerebroventricularly to the animals. This was done in rats, and we collected the brain tissue 24 hours after the administration to perform analysis of inflammatory markers and look potentially at the NF-kB signaling pathway. And obviously, we had a group receiving the PPAR gamma agonist, rosiglitazone or pioglitazone, and we compared this to control groups with saline, ICV administration, and vehicle IP, so the agonist was administered intraperitoneally. So here is some of these data in vivo where we observe upregulation of a number of pro-inflammatory cytokines, TNF-alpha, IL-1 beta. This was at the mRNA level, at the level of the hippocampus, and we see that in the presence of rosiglitazone in that group, we could mitigate the upregulation of these inflammatory cytokines. This was nicely reversed in the group receiving the agonist plus the GW compound, a specific antagonist, potent antagonist of PPAR gamma. So this was just performed to document the specificity of the effect that we observed. We looked also at the glutamate transporter, the GLUT1. In this case, we showed a trend in the GP120 injected animals to downregulation. However, we looked also at the protein level, and we saw significant downregulation of the GLUT1. And we saw this in vivo, and we saw it also in vitro in studies performed in primary cultures of astrocytes. So obviously, our target is the PPAR gamma. Part of our hypothesis is that in the context of infection, we will see a downregulation of this transcription factor of this receptor. And indeed, this is what we observe in the 3 AMD animals that were exposed to GP120. This we demonstrated also not only at the mRNA level, but also it was quite robustly decreased also at the protein level. We moved on into looking about potential mechanism of the PPAR gamma through the potential inhibition of the NF-kB pathway. What we observed, so what we did the studies is in vivo, after collecting the brain and isolating the hippocampus, we followed up in doing nuclear extraction from the hippocampal tissues and looked at the phosphorylated form of the subunit P65. As expected, we observed an upregulation of the P65 phosphorylated form. However, in the group and treated with rosiglitazone, this was reduced and mitigated. So potentially going along with the proposed mechanism, one of the proposed mechanism by which PPAR gamma exerts its effect in terms of the anti-inflammatory responses that we have observed. So in summary, as I said, I could not show all of the data. This was published a number of years back in the Journal of Neuroinflammation, is that we investigated PPAR gamma in this initial study as a potential target for HIV-associated brain inflammation using this very simplified model of only administering one viral protein, the viral code protein GP120. The initial data from the in vitro and in vivo acute study demonstrated brain inflammation responses and showed an anti-inflammatory potential of the PPAR gamma agonist, possibly through inhibition of the NF-kB pathway. So these initial studies were encouraging. However, they presented a number of challenges and limitations, as the rodents, as I was mentioning, were only exposed to only one viral protein, and there are many other HIV proteins, and we were reproached in grant reviews and also in our manuscript by reviewers, obviously, that our model was very limited and not that representative of the clinic. We also have to recognize the limitation of one single injection, and we measure response only over 24 hours, so an acute response. So these obviously poses the challenges that when we want to investigate treatment strategies and look at behavioral impairment and potentially reversal of those impairments, that our model could not allow for this. So in search of a more representative in vivo small animal model of HIV, we came across with the EcoHIV, and we were very fortunate that we could establish a very nice and productive collaboration with David Volsky and his laboratory at the Icahn School of Medicine in New York. So what is EcoHIV? I'm sure many of you attending know very well and work with this model. So very rapidly, it's a chimeric virus based on the backbone of HIV-1 and DK, where actually the viral HIV code protein GP120 is replaced with the ecotropic murine leukemia virus GP80. So GP80 now is able to permit the entry of the virus into mice, and this is facilitated by the murine cationic amino acid transporters. So these provide us here a really nice model that is infective for the mice, but not for humans, and that is convenient to work with. So the EcoHIV model has been well characterized by the Volsky's laboratory, demonstrating long-term infections of target cells, not only CD4 positive T cells, but also myeloid cells, microglia, and macrophages. They demonstrated clearly inflammatory responses along with neuronal damage, and a number of groups also demonstrated that this model displays neurocognitive impairments that reflect the ones seen in the clinic. So we went ahead and got the plasmid, the EcoHIV plasmid from the Volsky's laboratory. We produced the virus in our lab and started with working with this model. So I'm just going to walk you through our different studies over the years. We started this pre-COVID, and the work was interrupted, and then we picked up a few years ago. So the initial studies were performed in the mice administered intracerebroventricularly the EcoHIV, about 1 million picograms of P24. We did studies at 5 days, at 12 days, 21 days, and more recently up to 40 days to be able to do the behavioral work. At the different times of infectivity, we collected the tissue, the brain tissue. In some instances, we collected subcortical tissue, and in others, different regions, brain cortex or hippocampus, and other areas, and measure cytokine, chemokines, viral infection, and more recently performing behavioral studies. And obviously, we looked at the effect of the PPAR gamma agonist administered intraperitoneally using rosiglitazone or pioglitazones. So here are some data from these initial experiments. These were 5 days experiments looking at infectivity, measuring TAT. We demonstrated high viral RNA copies of TAT in the EcoHIV mice. This was reduced significantly in the presence of the rosiglitazone in the group receiving these agonist rosi or pioglitazone. We also looked at the capsid protein at P24, and we also demonstrated high levels of P24 in the hippocampus and mitigation, again, reduction in the presence of pio or rosiglitazone. So this was encouraging data. We moved on into measuring inflammatory markers, and again at 5 days post-EcoHIV administration, and we see here the data for TNF-alpha, L1 beta, interferon gamma, and we observe robust inflammatory responses in the hippocampus, and this was mitigated in the presence of pioglitazone. And again, we demonstrated specificity of the effect in the presence of the antagonist. We measure also number of chemokines, CCL2, CCL3, and CXCL10, and again, similarly, very robust upregulation of these in the infected hippocampus samples and mitigation in the presence of the treatment. And I cannot present all of the data to move on in more recent studies, but in the initial studies of the EcoHIV that we performed, so we implemented the model, and obviously it's robust model providing inflammation, cost-effective. So this was very attractive, easy to work with. We demonstrated anti-inflammatory and antiviral effects of these PPAR gamma agonists, and we demonstrated this in vitro and in vivo. However, the use of these agonists, as probably many of you know, I mean, they are used in the clinic, but their use is limited by their common side effects. There is an issue of safety with these compounds due to problems of fluid retention, cardiovascular effects, as well as weight gain. So from there, what question that we address, could we look at some novel compounds that are being developed that are PPAR gamma agonists? And we came across this compound, the INT-131. This is a selective PPAR gamma modulator, or SPARM gamma, that showed very effective anti-diabetic responses in patients with diabetes type 2. So this compound is in clinical trials with a high safety profile. So we got interested by this compound. We couldn't find anything in terms of neurodegenerative disorders, only one study on MS, but that we never could see a follow-up to it where they were looking into a potential neuroprotective effect. So what is INT-131? It's specifically designed to retain anti-diabetic properties of PPAR gamma full agonists while eliminated the side effects. It's chemically structurally different from the thiazolidinones, such as rosi and pioglitazone, and it also interacts through a distinct binding sites, which results in differential gene expression regulation and partial activation of the receptor. So our studies as pilots started by looking at the biodistribution of the INT-131. There was nothing that we could find in the literature for this. We developed an LC-MS mass spec quantification assay to detect and quantify the compound in biological fluids and tissues. And we did pharmacokinetic studies after the administration of one single dose of INT-131 administered intraperitoneally. We did this over a time interval of 5 minutes to 12 hours. And we see here the plasma, the PK plasma profile over time, which demonstrate obviously a peak post-IP administration and a very rapid elimination. So very rapid clearance that we identified for this compound, a half-life of about 1.5 hours. So these concentrations in plasma appear to be quite high, and our interest obviously was to see, can this compound penetrate into the brain? And we observe brain concentrations that were much lower, obviously, than in the plasma, not surprising, but the compound could permeate into the brain to plasma concentration ratios are still remaining quite low. So this was administration intraperitoneally. For the plan of our studies was to do studies over a long period of times. So practically and according to animal ethics and animal well-being, we cannot administer IP for a very long time. So we needed to move to the oral administration. So for that, because oral gavage is difficult to do and becomes tedious over a number of weeks, of days and weeks to do, what we did is that we prepared this compound in a pellet of peanut butter. And obviously rodents love peanut butter, and we know from other studies that the adherence is 100%. They will eat the pellet as soon as it will be offered to them. So these allow us to do a number of initial studies looking at the potential of INT-131 over a long period of time to mitigate and reduce the inflammatory responses that we observe in this EcoHIV mouse model. However, we faced another challenge, is that when we wanted to do the behavioral studies, we had to move to a different facility that was a peanut-free environment. So we could not bring our pellets to this facility. So that brought us to find ways of administering our compound to the animals over a long period of times. And we use osmotic pumps that we implant peritoneally that are loaded with our compound and that deliver 20 milligrams per kilo per day. So again, we came back and did a pilot study looking at the biodistribution of the administration of the compound via osmotic pumps over a long period of time. This was for 28 days. That's the maximum that we can deliver via these pumps. And our data actually showed very comparable, very reasonable result, comparable to the oral or IP administration of the INT-131. Our interest again was to look at the brain, and the brain concentrations remain low, but we could still detect these concentrations in the brain over the course of the time course of the 28 days via the osmotic pump administration. So now we have a way of administering our compound over a long period of time. So we took our animals, so we had 4 groups. Animals administered saline or only the INT-131, or infected with EcoHIV, or infected with EcoHIV plus our compound. Day 0 is an administration of EcoHIV, so it's the day of the infection. So here now in these more recent studies, we move from the intracranial EcoHIV administration to an intraperitoneal administration, just to mimic the clinic better. So this is a systemic infection. And 2 weeks after the infection, we implanted the osmotic pumps in the animals. And then we followed up after with behavioral testing. And at 42 days, we euthanized our animals at the completion of the behavioral testing and collected brain and spleen tissues, as well as plasma. And we proceeded primarily with, for now, with qPCR analysis to assess a number of inflammatory markers, blood-brain barrier markers, oxidative stress. We would like to look at apoptosis and, of course, infectivity. So here are some of the data in terms of infectivity. I'm showing here data for VIF and TAT. In spleen, we observe here, as anticipated, increase in the viral RNA copies for both, and we observe a mitigation and of the infectivity in the group receiving the INT-131 for the 2 viral proteins. Similar data observed in brain cortex, although the number of copies, as anticipated, because this was a systemic infection, were lower in the brain for both. And again, mitigation and reduction of the effect in the presence of INT-131. Inflammatory responses, I'm showing here in spleen, the IL-1 beta, TNF alpha, and the chemokine CCL2. So we observe some modest upregulation of these inflammatory markers. And in the brain, similar data was observed. And this was interesting that via the IP administration, we could still detect these effects in the brain, which we wanted. So blood-brain barrier dysfunction is another aspect of high interest to that in terms of leakiness of the blood-brain barrier and dysregulation of tight junction proteins. So we looked at TGP1, this is the ZO protein, occludin, and claudin-5, these tight junction proteins. They were all downregulated in the EcoHIV samples, and there was mitigation of the effect in the presence of the treatment. And we also performed a permeability study using sodium fluorescein, looking at the permeability of sodium fluorescein into the brain. So what we observe is increased permeability as anticipated in the EcoHIV mouse model, and this was nicely mitigated in the EcoHIV infected group receiving the INT-131. PPAR gamma is our target, so we wanted to look at PPAR gamma. We observe a downregulation. We had observed this before when we administered the GP120, and we observe restoration of the levels in the presence of INT131. So this is quite encouraging. And we are following up the work here with immunohistochemistry to look at more at the protein level. So before I close up with the summary, what I would like to mention is that at present, we are following up with a number of behavioral studies. So we are doing these studies in males, but also in females, in a separate group of females. We feel it's important to address sex differences. We couldn't find very much in the literature addressing this aspect for EcoHIV. It's possible that it's out there. We couldn't potentially see it. So we are still in the process of analyzing those data, particularly for the female. So I have not brought it in here, but we are doing extensive behavioral testing, looking at locomotion, looking at anxiety, applying the open field test. We are looking at novel object recognition. So we are in the process of analyzing those data at the moment. So in summary, the treatment with this selective PPAR gamma partial agonist, INT131, reduce EcoHIV mediated infectivity and inflammatory responses. This we've seen in vivo in the EcoHIV mouse model. I want to mention that we did also a number of in vitro studies exposed to EcoHIV in primary cultures of mixed microglia and astrocytes, which I didn't go over or time to present. From the initial behavioral studies that we've done in males, we are seeing that the compound, first we are seeing and demonstrating that in our own hands, the EcoHIV model is showing deficit. Particularly, this was detected in the novel object recognition that we have performed already in males, and that the INT131 could alleviate and reverse some of these deficits. We have characterized the compound pharmacokinetic biodistribution profile and demonstrated that it can reasonably enter the brain. And as most recent studies, the INT131 appears to improve learning and memory performance in the model. So overall, our data provides evidence that this compound could be a potential candidate for the treatment of hand or NCI. The next steps are to complete the studies in the female mice, comparing them to the males, both for the behavioral tests that we have been performing and are in the process of analyzing, as well as for the inflammatory responses and the infectivity. Following that, we are thinking that already based on this data that could be of importance to follow up and testing this compound potentially in an SIV-infected non-simian model, non-human primate model, potentially before bringing it to the clinic. So I want to close by acknowledging a number of members of my group, the contributing members to this work, in particular Celine Titus that we see here, and Toza Melhawk. Oops, sorry. And older group is here. This was just pre-COVID, Amila Omeragic, who initiated the work and implemented the EcoHIV mouse model, and Tozamel Hawk is here. I want to thank our collaborators, in particular David Volsky and members of his lab, such as Jennifer Kelschenbach, for their tremendous help in the implementation of the EcoHIV model and follow-up work that we've done, as well as a number of members of our department that have contributed, particularly in the advisory committee meetings of the doctoral candidates. I also want to thank the Center for Pharmaceutical Oncology, Azal Marouki, for the help that she provides in training our students with major equipment, such as the mass spectrum photometer, imaging equipment, etc. So she provided help with the quantification analysis by LCMS. Funding agencies, I want to acknowledge the Canadian Institute of Health Research, as well as the Canadian Foundation for AIDS Research, for supporting this work since its inception, as well as the CanCure and particularly the Ontario HIV Treatment Network and the Center for Pharmaceutical Oncology. So thank you very much for being here, for attending, and I'm happy to address any questions that you will have.
Thank you, Dr. Bendayan. That was really nice talk. So let me first go with the question in the chat panel. Then we're gonna get to those people raising a hand. Dr. Rice, are you around?
Hi, thank you, Reina, for a wonderful talk. Just wanted to know if the initial thiazolidine specifically rosiglitazone, was shown to penetrate the BBB.
Yes.
And when I was looking in literature, I found it's not very brain penetrable. So were the concentrations matched, or do you think it was? So it was.
Yes, we looked into that. I didn't have the chance to present it. We analyzed the levels through, again, mass spectral CMS mass spec, and it is low, but they are able to permeate into the brain. I also want to mention that for the INT131, because of our kind of area, more our expertise in transporters, that we obviously looked into to see if this compound is a substrate for ATP binding cassette membrane transporters, and indeed it is. But still there are modest amounts, as you could see from our studies, that can permeate and that can have an effect. And for that, I mean, obviously, if the compound is administered orally, we had to obviously go to much higher doses as well, due to aspects of bioavailability, etc. So because this compound is fairly safe, we also feel that in having it in the oral form, obviously we had to go higher, but we are still within we are still within the safe range of doses that have been used for the treatment of diabetes.
Thank you.
Right.
Thank you so much.
Dr. Kumar?
Yeah, thank you, Reina. This is a very impressive talk. My question is about when you looked at these viral mRNA and also cytokine, chemokine mRNA in different regions of brain, right? Have you also checked like whole brain? Is it detectable if you do the whole brain, or also in the plasma? Can we detect those, either protein or something?
Okay, so good question. The answer is no. We haven't. I'm trying to think because we've done so many different studies. We haven't done whole brain. We have either taken the subcortical region when we did the intracranial administration of EcoHIV. We work primarily with subcortical. In the IP administration of EcoHIV, it's a good point. We haven't yet looked at plasma, and we should. we looked at spleen and brain, and in those studies, we are looking at different brain regions like brain cortex.
Okay, thank you.
Yeah, no, that's an important aspect. Thank you.
All right, Dr. Pogrinsky.
Yes, thank you. Thank you for a nice talk. Very interesting. I have a related question to the previous one. So basically, you showed TAT expression in the cortex. Did you analyze any other regions like of the brain to see whether there is any kind of expression of viral proteins? And then another related question is, since the mice are immunocompetent, and it is well described that that EcoHIV replication actually is maintained for a very short time and then completely suppressed. So I just wonder whether you measured it, you showed only one time point, whether you measured it at different time points to see how it changes. And related question is, don't you think that because EcoHIV is suppressed, that and basically to develop new behavioral impairment, you just need quite a long time, as we know, that basically you won't be able to really get a nice impairment to detect anything with this model.
So from our studies performed at the 42 days, in terms of behavioral impairment, we are seeing in male mice and now in female that there is cognitive deficit, and that we can reverse this with the administration of the compound. Because we are still in the process of analyzing the data, I didn't bring it in. We are putting it together because the female study took us, obviously it's a separate study that we have recently collected the samples. So we are looking into all of the analysis. So I guess that your point is well taken, but we have looked at infectivity at different time points. We've looked at infectivity at 5 days in the EcoHIV model. We are looking now at the infectivity at a much longer period of time in both in spleen and in the brain. And it looks like we are, from our data, we are detecting higher copies of these viruses. So we are looking at brain cortex, but we have other regions that we would like to analyze as well.
Thank you.
Right. So we are right on time. Any other question, by the way?
Yeah, I have one.
Oh, here,
question.
go ahead.
Okay. So your experiment approach is really great, especially the pharmacokinetics approach to measure the efficacy of the brain barrier. I have questions like, you told about PPAR also has neuroprotective effect, but you did not see, I did not see like, anything. Are you planning or already you have done with spine kind of degenerations or regenerations or kind of synapses, more formations, kind of neuroprotection? How you will explain that there are neuroprotections, in vivo or in vitro after the treatment?
Yeah. So that's an important question. We haven't done those type of experiments, and those are important. In the plan of what we would like to do, we would like to look at a potential apoptosis through measuring caspase. We would like also to look at the MAP2 because that will show us potentially, aspects in terms of the microtubule defects, right? But your point is well taken. We haven't done, we still have the samples. We haven't analyzed those markers.
Okay. So another one, just a simple, you talk about motor behavior. Are you also focusing on measuring the motor behavioral studies to correlate the efficacy of the drug, like, which you want to, like INT 131?
Yes, we would like to also do motor.
So
We would
motor-
like to do motor, we would like to do locomotion as well. Yeah.
Okay, so like, you want, because there are not only cognitive impairment happens, there are so many
Yes.
motor impairment happen. So are you
Yeah.
focusing on nigrostriatal circuit related motor impairment, or kind of in general locomotions, like in a complex way or simple way, specific targeted for related to Parkinson kind of
Yeah, that's a good point. I think there we are going to look at a more simple locomotion. I believe. Yeah.
Okay. Thank you
Yeah.
very much.
Yeah, no problem. Yeah, but those aspects are, quite important in terms of ongoing behavioral work.
All right. So Caroline, can you tell us about the upcoming events?
Sure. So we will not have a seminar next week due to
Okay.
the Thanksgiving holiday,
Mhm.
but we will have a seminar on December 4th. It's Aaron Schorer, and he is speaking about divergent metabolic profiles in NeuroHIV, signatures of cognitive and motor impairment.
All right. Thanks, Caroline. Oh, and thanks again, Dr. Bendayan. That was a
you.
fantastic talk. And everybody has a fantastic Thanksgiving weekend. And we'll see you 2 weeks from now. All right. Have a nice day.
Thank you
you.
very much.
Mhm.
Pleasure of
Thanks.
interacting once more.
Me too.
Bye.
Bye-bye.
Bye, Rana.
Thank you.